Hello everybody, my name is Nicole DeLong, and I'm a current master's student at the Fraunhofer Institute for Algorithms and Scientific Computing. I want to talk to you today about my work finding novel COVID-19 drug targets based on a network representation learning approach called guilty targets. Although we have vaccines developed for COVID-19, there's still an interest in finding drugs for perhaps attenuating COVID symptoms or reducing the severity of the disease for those who still get it. Now, when I refer to drug targets, I'm referring to proteins which we can interfere with drugs in order to achieve some of these goals. To find novel drug targets, I use what's called the guilty targets workflow. This workflow was published in 2020 and requires 3 forms of input. The first is a network containing protein-protein interactions, where every node represents a protein and the edges between them represent the interactions. The second is a list of known potential drug targets against COVID-19. Each node in the graph will be labeled as a potential drug target or unlabeled. And lastly, I used differential gene expression data for either cells or patients infected with COVID or the SARS-CoV-2 virus versus control. Now, the guilty targets workflow takes a series of random walks through the network and collects information about structural contexts and attribute contexts. These serve as the input for a skipgram model, and this skipgram model then creates low-dimensional representations or embeddings for every node in the network. These embeddings serve as input for a binary logistic regression classifier, and this classifier's job is to discriminate between the known potential drug targets and the unlabeled nodes in the network. Now, obviously, there will be some false positives which are classified among the known potential drug targets. And these false positives serve as novel predictions for drug targets. In addition, the beauty of a logistic regression classifier is that it not only produces a binary classification, but also a likelihood score of each node or protein in the network of being a drug target. And we can use that score to rank every protein in the network as a potential drug target. So I ran this workflow on a series of different types of RNA-seq datasets. I first used bulk RNA-seq datasets, which I got from the Gene Expression Omnibus, and I additionally ran it on single-cell RNA-seq datasets based on the cell type. I got the single-cell RNA-seq datasets from the Gene Expression Omnibus, as well as Yale Medical School and Charité Medical University of Berlin. Here we have a visualization that's sort of like a Venn diagram in the way that we can see the overlapping results, but in a different way. So for the 6 bulk RNA-seq datasets, we can see that the greatest group of overlapping top hits that resulted from the guilty targets workflow are actually shared among all 6 datasets. And this is reassuring because it shows that there's consistency in this algorithm. You can also see here that the next largest groups of overlaps were actually distinct for individual data sets. And that shows that this guilty targets workflow also has some sort of data set dependency, which further reinforces the idea that one should use multiple data sets when trying to do something like this. We can see the same sort of thing in the single-cell RNA-seq data, which, as I said, I separated into different cell types. The largest group is shared among all cell types, whereas, again, the next largest groups are distinct for individual cell types. This is also interesting because we can see sort of cell type signatures here, based on the top target hits that were found. From these top hits, I used some filtering steps and compiled a list of top hits which were consistently found across multiple data sets. So On the left here, you can see that 2 targets, AKT3 and PRKG1, are potent drug targets for 2 drugs, mirimistat and prenomistat. In a recent Nature paper, these drugs were found to significantly inhibit SARS-CoV-2 replication, which experimentally validates them as likely potential drug targets for COVID. Additionally, a collaborator found that drugs which were potent against MAP2K7, GRK2, and CBSL significantly reduced cytopathic effect of SARS-CoV-2 in VEROE6 cells. So we have 5 of these top hits found by the Guilty Targets workflow that were experimentally validated. I took this experimentally validated group of targets and ran a gene ontology enrichment analysis on them. I found that among biological processes which were positively associated with this set of targets, one of those was viral entry into the host cell, which further shows that these drug targets could be involved in viral processes, especially SARS-CoV-2 involved processes. So, in conclusion, I used what's called the Guilty Targets approach to predict novel drug targets and found that 5 drug targets, MAP2K7, GRK2, CBSL, AKT3, and PRKG1, are experimentally validated. Here are some funding and references that I'd like to acknowledge, and if you'd like to contact me, feel free. Thank you very much.